And you can go ahead, Carol.
Okay, thank you. Good morning, everyone. I'm really pleased to have our fabulous senior scientist postdocs here today, giving 2 short talks for the Cancer Center. And what I'd like to do is kind of tell you a little bit about each first, and so that they can just get right into their talks, leaving time for questions after each talk. Our first speaker will be Dr. Thu Truong. Thu received her PhD in chemistry and biochemistry in Kate Carroll's laboratory at the University of Michigan and the Scripps Research Institute. She has joined my lab for her postdoctoral studies and has been in my lab, how long? Maybe 4 years? Are we at the 4-year or the 5-year mark? I can't remember.
We're actually at the 7-year mark.
Oh, not in my lab. So anyway, Thu has done phenomenally well. She had several high impact papers from her PhD studies. She has had several really nice papers from my lab in collaboration with Julie Ostrander, where we've been senior co-authors. She's won a number of competitive awards. She's an AACR scholar. She is a University of Minnesota Postdoctoral Career Development Awardee. She's won that twice. She's won numerous awards from the Endocrine Society for her research and research presentations. And she was also awarded an Early Career Development Award from Metaviver. And she also has an award letter on her desk from the NIH. She's got a K-22. And she has been a terrific role model for my trainees, especially young women trainees. She is a go-getter and phenomenal in the lab. And I think you'll see that in her research. And similarly, Kiel Tietz is a postdoc in Scott Dehm's lab, and he has been on the T32 fellowship in cancer biology. He received his PhD from Ohio State University and in the program of molecular cellular and developmental biology, where he studied squamous cell carcinoma. He has joined Scott Dehm's lab, where he works on messenger RNA regulation, turnover, and its role in gene expression and in prostate cancer. He also is a phenomenal scientist. He is the awardee of a DoD Early Investigator Research Award. Only 16 of these were given in the world. 2 of them have come to our university, and Kiel has one. And so I'm just so proud of these postdocs. I'm so impressed. We're in 2 years of COVID. They haven't been able to go to meetings. They've faced the Sunrise Plan. They faced the subsequent supply issues like all of us have, but they've still managed to rise to the top. And I think this shows true grit, dedication, fortitude, and a real, you can see the enthusiasm for doing research, and through all this adversity that they've been able to further their careers in such a really outstanding way. And so with that, I'll let Thu begin her talk, and then that will be followed by some questions. And then Kiel will proceed with his talk. And again, thanks again, you guys, for doing this. This is going to be great. Go ahead, Thu.
All right, thanks for that, Carol. So today I'm going to be talking about some of the work that we've done to understand therapy resistance in the context of cancer stem cells in ER-positive breast cancer. It's not working. Okay. So clinically, breast cancers are divided into different molecular subtypes based on the expression of 3 receptors, and those are HER2, estrogen receptor, and progesterone receptor. The luminal or ER-positive subtype accounts for a majority of cases, and this subtype is defined by the presence of ER and contains a range of PR-positive cells as well. And expression of these receptors is important because treatment options are largely dependent on exploiting their activity. So from the survival curve here, you can see that ER-positive tumors tend to relapse late compared to other subtypes. And what I mean by that is recurrence that occurs more than 5 years after diagnosis. So for example, the triple negative subtype has a sharp decline initially at 3 to 5 years after diagnosis, whereas the ER-positive subtype, and that's the orange line over here, has a more gradual decrease over time. And ER-positive patients tend to respond very well to endocrine therapies initially, but up to 40% of patients will eventually develop resistance that can progress to metastasis. Contributing factors to late recurrence and metastatic spread include cancer stem cells, and these cells tend to be poorly proliferative and can exist as minority populations in resistant tumors. Their ability to survive treatments enables them to escape standard therapies that are aimed at targeting rapidly dividing tumor cells. And then in line with this, I also want to highlight some other resistant mechanisms, and those include deregulation of co-regulators and altered metabolism, all of which we believe in the context of our work contribute to driving cancer stem cell survival and expansion. And that brings me to the key players of our story, steroid receptor coactivators PELP and SRC3, and how they cooperate in ER-positive breast cancer. So PELP was first identified as an ER coactivator, and it's primarily found in the nucleus in normal breast tissue, where it acts as a co-regulator that's involved in chromatin remodeling and RNA processing. PELP can also dynamically shuttle to the cytoplasm, where it serves as a scaffolding protein for growth factor and steroid receptor pathways. Now, PELP is found to be overexpressed in a number of endocrine cancers, and those include endometrial, ovarian, and prostate. It's overexpressed in more than 80% of breast tumors. And then altered localization of PELP to the cytoplasm has been seen in up to 50% of PELP-positive tumors. Now, because of this, cytoplasmic PELP has emerged as a marker of increased breast cancer risk and also aggressive tumor behavior. Now, in a study that looked at the correlation between cytoplasmic PELP levels and response to tamoxifen treatment, so tamoxifen is an endocrine therapy, they found that low cytoplasmic PELP levels were correlated, low cytoplasmic PELP levels, patients that had that benefited more from tamoxifen treatment, whereas those with high cytoplasmic PELP levels had a poorer response, suggesting that when PELP becomes more oncogenic, when a larger fraction of it is retained in the cytoplasm. Now, taking the studies from the literature into account, the goal of our own studies was to really get at the mechanisms that promote oncogenic PELP signaling in ER-positive breast cancer. And in order to do so, we sought to identify interacting partners that were unique to PELP in the hopes that we would identify some that were distinct to the cytoplasmic compartment. We used a tap-tag mass spectrometry approach to identify binding partners specific to PELP. And so what we did was we infected HMAX with adenovirus containing tap-tag PELP, affinity purified the samples, and then analyzed unique bands for the cytoplasmic and nuclear extracts by mass spectrometry. So on the left here, in the nuclear extract, many of the bands that we identified corresponded to proteins that were involved in RNA processing, which, if you recall, is a known nuclear function of PELP. And then on the right, in the cytoplasmic extract, the band of most interest to us corresponded to a protein known as SRC3. And this is also known as AAB1 in the literature, which stands for amplified in breast cancer. So SRC3 is a nuclear receptor coactivator, and its gene locus is amplified by about 10% in breast and ovarian cancers, and it's overexpressed in 30 to 60% of breast tumors. SRC3 has to be phosphorylated in the cytoplasm in order to induce nuclear translocation and ultimately its coactivator function. And additionally, increased cytoplasmic localization of SRC3 has been seen in breast tumor samples from IHC staining. And so I just want to note that a lot of studies have shown that PELP and SRC3 contribute to cancer cell biology independently of each other. But what's important to note is that they do have several functional parallels, such as both of them being involved in mediating proliferation, migration, and contributing to endocrine resistance. And that's really why SRC3 jumped out at us initially when we were trying to determine which leads to follow up on from our TAP pull-down studies. So to study signaling in the context of cytoplasmic PELP, we generated models in MCF7 cells. And this cell line contains high endogenous levels of PELP, which also directly correlate to high SRC3 levels, making it an ideal cell line to study both of these coactivators in. So what we did was we knocked down PELP and then re-expressed either wild type or cytoplasmic PELP by mutating the nuclear localization signal as shown here. And we call this mutation cytopelp for short, and this approach has been used by our lab and other labs in order to slow the rate of nuclear translocation and ultimately increase steady state levels of PELP in the cytoplasm. We did a cellular fractionation, and it shows that our cell line models are behaving as we would expect them to. So wild type PELP expressing cells contain a larger fraction of PELP in the nuclear extract, whereas the cytopelp cells contain a larger fraction of PELP in the cytoplasmic extract. And then over here on the right, I just have some immunofluorescent images showing the localization of PELP in our cell line models, which confirms our fractionation results. And then in the panel above is PELP IHC staining from normal mammary gland, and then 2 examples of invasive breast cancer, indicating that our cell line models do behave, do reflect that increased cytoplasmic PELP that is seen in human breast tumor samples. So we see that in our cell line models, PELP preferentially interacts with SRC3 when it's in the cytoplasm, and additionally ER and PR also preferentially bind to cytoplasmic PELP, and its expression also has an impact on the ER SRC3 interaction as well. And this was all done using co-immunoprecipitation assays. So together, these results show us that PELP is involved in altered signaling with steroid receptors and their co-activators in the context of the cytoplasm. Now, I alluded to some of the previous literature that has shown that cytoplasmic PELP is involved in mediating proliferation in ER-positive breast cancer, and we've also shown that in our models. And I want to note that proliferative and cancer stem cell programs are mediated by different pathways. And so what we did was we took our cell line models and performed tumor sphere assays with them. And tumor sphere assays are an assay to measure stemness and progenitor activity, and those are performed in 3D culture conditions. And we see that in the cytopelp expressing cells, these have increased ability to form tumor spheres relative to the vector control and the wild type PELP cells. And then to extend our tumor sphere findings, we also measured known breast cancer stem cell markers, and those would include aldehyde dehydrogenase, so ALDH activity, and then also the CD44 high, CD24 low population. And we see that both of these breast cancer stem cell markers are increased in the cytopelp cells. When we knock down SRC3, we see that this reduces tumor sphere formation in our MCF7 PELP models. And then in addition to the knockdown studies, we also utilize SI2, which is a small molecule inhibitor of SRC3. We see that with treatment with SI2, that reduces the PELP SRC3 interaction from forming and also reduces tumor sphere formation in the MCF7 PELP models, indicating to us that SRC3 is a key mediator of PELP-induced cancer stem cell populations. So just to wrap up the beginning part of this talk, we've demonstrated that PELP is involved in altered signaling within the cytoplasm and forms a complex with another coactivator, SRC3. This complex promotes breast cancer stem cell phenotypes. And when we use either small molecule inhibitors or genetic knockdown approaches, we're able to disrupt this coactivator complex from forming and ultimately any CSC populations that arise from it. So the findings from our first study kind of naturally led us to wonder, how can we truly target these cancer stem cell populations? Progress in treating metastatic ER-positive breast cancer has been limited by therapies that are aimed at targeting rapidly proliferating tumor cells. So, how can we use what we know in order to take advantage of the slow-growing nature of the CSC population, particularly in the context of therapy resistance? And so that was the goal in our next line of studies. We wanted to further define how these coactivator complexes act as key mediators of signaling pathways that contribute to CSC-driven therapy resistance. So I've mentioned that breast cancer stem cells are a minority population, and you can imagine that this makes it a little bit difficult to detect specific changes in gene expression. And so if you look at panels A and B over here, we see it's really, when we culture the MCF7 PELP cells in 2D adherent or 3D tumor sphere conditions, we see that it's really those 3D conditions that drive the increase in ALDH activity and also the CD44 high, CD24 low population. And so based on these comparisons, we know that the 3D culture conditions really enrich for the CSC population. So what we did next was an RNA-seq study in order to be able to identify cytopelp-specific regulated pathways and also to distinguish any differences between 2D and 3D culture. And so the results of that RNA-seq study are summarized over here on the right in panel D. And so we did a 2D versus 3D comparison, and then we took the cytopelp up and down regulated genes, and using those genes, we performed pathway analysis. So from our IPA pathway analysis, 3D cultured cytopelp cells were predicted to upregulate HIF-activated pathways, particularly those involved in cellular metabolism. And then over here on the right, I just have sort of a representative heat map. And this heat map was generated from the genes that were grouped in with the HIF-activated and the ATF4 pathway. And the red arrows are pointing to genes of interest to us. And then we ultimately decided to focus on Pfkfbs, which are metabolic bifunctional kinase phosphatases that are involved in glycolysis and have known roles in cancer biology. We validated a few select target genes from our RNA-seq by qPCR, and we see that mRNA levels of Pfkfb1, so that's the gene name for HIF2, Pfkb3 and Pfkb4 are upregulated in the cytopelp expressing cells compared to the LX7 vector control and wild-type PELP, particularly in the 3D context. Now, given the central role of HIFs in cellular metabolism, we started to wonder if PELP localization would have any effect on mediating altered metabolic pathways. And so in order to assess this, we performed Seahorse cell energy phenotype tests, which allows you to measure oxygen consumption rates and extracellular acidification rates. So I'm going to refer to those as OCAR and ECAR. And what we see is that the wild type PELP cells have increased OCAR and ECAR levels relative to the vector control, and that trend is further enhanced in the cytopelp expressing cells. So these data from this slide and then the previous slide show us that 3D culture cytopelp cells upregulate HIF-activated gene programs that include Pfkb3 and Pfkb4, and that PELP localization appears to have an impact on mitochondrial respiration and glycolysis, indicating that there's probably some plasticity at play here. We also hypothesized that the Pfkfbs would be components of the PELP-SRC3 complex, and we do see that there's an increased interaction through co-IP assays between PELP and Pfkb3 and PELP and Pfkb4. Treatment with PFK158, which is an inhibitor of Pfkfb3, and then 5-MPN, which is an inhibitor of Pfkb4, reduces that PELP-SRC3 complex from forming. And we also utilize a proximity ligand assay, which is another method to measure the protein-protein interaction in the cell. And we see that our PLA results also reflect what we saw in the co-IP assays, that the PELP-SRC3 interaction is reduced in both wild type and cytopelp cells when you treat with the Pfkfb inhibitors. Naturally, we also looked at the effect of the Pfkfb inhibitors on tumor sphere formation, and we see that in both cases of the inhibitors, we see a reduction in tumor sphere formation in our MCF7 PELP models. Now, to extend and complement our in vitro studies, we also performed in vivo studies using the mammary intraductal xenograft model. And so I'm going to use this schematic to kind of explain why we chose to go with this approach over traditional approaches. So in traditional orthotopic approaches, so on the right, or sorry, on the left here, the cells are injected subcutaneously into the mammary fat pad. And this has just not been shown to be a good model to model metastasis in when you're studying ER-positive breast cancer. And that is mainly due to the rapid growth of the primary tumor, because the mice usually end up needing to be euthanized before metastatic lesions can even occur. Now, into the intraductal approach, the cells are injected directly through the nipple into the ductal structure. And this has been shown to reliably form metastasis using ER-positive breast cancer cell line models such as T470 and MCS7s, and those occur on a 3 to 6 month time frame. So up until this point, Pfkb3 and Pfkb4 have both shown promising results in our cell-based assays. And so what we did was we wanted to look at the correlation between mRNA expression levels of the Pfkbs with overall survival. And we did this using the Metastatic data set. And so we see that there doesn't seem to be a correlation between Pfkb3 with respect to overall survival, but high Pfkb4 levels do seem to be associated with decreased overall survival in all subtypes of breast cancer and also the ER positive only cohort as well. So based on these results, we wanted to see if Pfkb4 knockdown would have any effect on primary tumor progression and or circulating tumor cells. And so that's where the introductal model comes in. So in order to do this experiment, we took MCF7 wild type and cytopelp cells containing either the GFP control or the Pfkb4 knockdown and injected them intraductally into adult female mice and allowed that to go for about 8 weeks. And we sacrificed the mice at those points, removed their mammary glands, and then did H&E staining and then calculated the percent tumor area from those H&E stains in order to determine primary tumor burden. And so let's focus on the GFP controls first. And we do see that the cytopelp intraductal GFP controls have a significantly increased primary tumor burden compared to the wild type GFP intraductal mice. And then when you introduce the Pfkb4 knockdown, it doesn't seem that the Pfkb4 knockdown is able to exert a significant effect. So PELP localization seems to be doing it, but not necessarily Pfkb4 knockdown. Another thing that we did was we also assessed the presence of circulating tumor cells. And we did this because CTCs are considered a marker of metastatic potential and also associated CSC behavior. And we did a cardiac puncture and drew blood at the same time that we euthanized the mice. And we took the blood samples, processed them, and then seeded them into soft agar assays. And then any cells that would grow out and form colonies in the soft agar assay, we would consider those to be to have CTC potential. So the results of that part of the experiment are kind of summarized on the bottom here in panel D. And so let's focus on the data that we obtain from the wild type mice first. You can see that in both the GFP control and the Pfkb4 knockdown, neither groups of those mice were able to form CTC colonies. But when you look at the cytopelp intraductal mice, starting with the GFP control, we see that those mice were able to form large and viable CTC colonies. And then when you introduce the Pfkb4 knockdown, that seems to reduce colony size, and also colony number as well. And that's quantified over here in panel F. So from this data, we made the conclusion that cytoplasmic PELP seems to drive primary tumor progression and also the expansion and formation of CTCs, the latter of which can be then attenuated by Pfkb4 knockdown. So I just want to take kind of a little break here to reflect on what I've shown you so far. We've been able to identify that Pfkfb kinases are important components of the PELP SRC3 complex. And then when we block its activity, we're able to block that PELP SRC3 coactivator complex from forming and also PELP-induced CSC and CTC populations. And so taking this data, we started to wonder, is the biology that we're seeing in our MCF7 PELP models applicable in chemotherapy or endocrine therapy resistant models, just given the fact that we know that PELP and SRC3 do independently contribute to tamoxifen resistance. And so in our next line of studies, we utilized a chemotherapy resistant model. So that was a paclitaxel resistance. I'm going to refer to that as Taxar. And then an endocrine therapy resistant model, so a tamoxifen resistant model referred to as TAMR. And so what we did was the first thing was we assessed the localization of endogenous PELP within the Taxar and the TAMR models. And what we find is that, when we culture the cells in 2D adherent or 3D matrigel conditions, and then we look at PELP localization from the immunofluorescent images, the resistant cell line contains a larger fraction of cytoplasmic PELP compared to the control. And that trend is further increased and enhanced in the 3D matrigel conditions, indicating that it's the 3D conditions that drive that dynamic shuttling of PELP to the cytoplasmic compartment. We also observed increased levels of the PELP SRC3 complex in both the Taxar and the TAMR lines compared to the control, and also increased mRNA levels of the Pfkbs in the resistant line compared to the control, particularly in the context of 3D culture conditions. We also looked at the effect of tumor sphere formation in these resistant cell lines in response to some of the inhibitors that we had been using. So the first thing I want to point out is that if you look at the basal tumor sphere levels between the resistant lines and the control line, both of those are increased in the two resistant lines compared to their respective control. And then when we treat with either SRC3 or Pfkfb inhibitors, we're able to reduce those tumor spheres in both the Taxol and the Tamar lines. We also looked at the effect of co-treatments in our resistant lines, and we thought that would be interesting to do because we wanted to know if any of the candidates that we identified from our study, could those be utilized to block the CSC population, and also if they could be combined with other standard of care treatments. And so one of the examples I'm showing you today is just an Si2-5-MPN combination. And the data for the Taxol cells is on the top, Tamar cells is on the bottom. And what we can see that in the Taxol cells, this co-treatment doesn't seem to be more effective than the single agent treatments. But then in the Tamar lines, and I know you actually can't see the labels on here, but this is the MCF7 Tamar lines, and this is the T47D Tamar lines. And we can see that this co-treatment does seem to be more effective than the single agent treatments in multiple cell line backgrounds. We also tested the effect of these co-treatments in ER-positive patient-derived organoid models that we obtained from the Whelm group at Huntsman Cancer Institute. So the first thing we did was we looked at expression levels of our key players in these organoid models, and we are able to detect PELP, SRC3, and the Pfkbs. And we wanted to see if the co-treatments in organoid models would have any effect in these tumor sphere assays. So what we did was—
You're a little bit over, maybe try to wrap that up pretty soon.
Yeah, I'm on my last slide.
Okay, awesome, thank you. Sorry, I had to interrupt.
The first co-treatment that we tried, I'm going to focus on, is the TAM-SI2. And we didn't see, any effect between the co-treatment and enhanced in the single agent treatment. But when we look at the data that we obtained with the Pfkb4 inhibitor, we do see that that does have an increased effect compared to the single agent control in multiple PDO models. And so just to wrap up the second half of the talk, we've demonstrated that PELP localization alters metabolic signaling in breast cancer cells through seahorse assays. Pfkfb inhibition blocks that coactivator complex from forming, and ultimately PELP-induced CSCs and CTCs. And we show that therapy-resistant models, such as a chemotherapy and endocrine therapy resistant, do phenocopy that PELP-SRC3 biology that drives that CSC phenotype. And finally, we demonstrate that endocrine therapies can be combined with PELP complex inhibitors. And so finally, I do want to thank my postdoc advisor, Carol Lang, and my longtime collaborator, Julie Ostrander, members of the lab that were involved in this study. So Kyla, Carlos, Betsy, and Ying, our collaborators, and then of course our funding resources.
Thanks, really nice, really nice. Are there questions? Those would come through the raise hand or the chat function. So one or two questions. There's a question.
Oh, that was you.
From Dr. Dehm, are the effects of SRC3 and Pfkfb targeting specific for stem cells, or are there effects on non-stem cells and or normal breast cells? That's a great question.
I guess we have looked at the effect of blocking that PELP-SRC3 interaction with the inhibitors in both. I would say that yes, we do see a reduction in the stem cells, but we also have performed assays in the not like kind of like the 2D adherent conditions, and we do see an effect. So like the co-IP assays are performed in adherent conditions.
Great, thank you. I don't see any other questions since we're just about 2 minutes behind, not terribly behind, and we did start late, so I think we're on time. So thank you to that was wonderful. And so now we'll hear from Kyle, and he's got his slides shared. Thanks, Kyle.
All right, let me just get my. All right, so I definitely just want to start by thanking the Masonic Cancer Center for allowing me to speak today. I am a postdoc in Scott Dehm's lab, and I'm going to talk about a project that I've been working on for the last couple of years, titled AR Alternative Polyadenylation as a Therapeutic Vulnerability in Prostate Cancer. And I just want to start this talk by just showing the prevalence of prostate cancer in our male population. This is actually a brand new Cancer Statistics 2022 slide. And you can see here the estimated new cases, 27% of new cancer cases in males are attributed to prostate cancer, and its second top estimated deaths to occur from male population by cancer is prostate. So it's extremely prevalent disease. And as we continue to have longer lifespans, it's only going to be becoming more and more prevalent disease in our population. I also just want to walk you guys through just a kind of a crude diagram showing typical prostate cancer progression and the treatment options that are given to patients. And so on the y-axis here, we just have disease burden. On the x-axis, we just have time. And so when a typical male develops prostate cancer, primary prostate cancer, the first line of defense is really surgery or radiation therapy to remove the primary prostate cancer. And luckily for most of the men, this effectively cures the disease and they're cured of prostate cancer. But unfortunately, a subset of these patients will have reemergence of this prostate cancer, and this disease burden will start to come up. And this is really this castration-sensitive prostate cancer phenotype that emerges. And kind of as the name denotes, the first line of defense for this is castration. It's removal of the source of androgens from these males, and that will help these males with the disease burden. It will start to go down. But unfortunately, it's just a matter of time until this castration-sensitive prostate cancer transitions to this castration-resistant prostate cancer, CRPC. It's an extremely aggressive prostate cancer that's responsible for virtually all the prostate cancer deaths. And I don't have time to go into it today, but it's been extensively shown that reactivation of the androgen receptor, its signaling is a critical driver of this phenotype. And so, males that develop this castration-resistant phenotype, the disease burden will start to go back up again. And then this is when next generation AR targeted therapy. So there's these really potent AR inhibitors such as enzalutamide. It's a very common drug given to men with CRPC. And this again will help these men. But unfortunately, it's just a matter of time until they develop resistance to even these AR targeted drugs. And ultimately the disease burden starts to rise very high and leads to the death of this patient. And so in Scott's lab, we're really interested in the drivers of this castration-resistant phenotype and better understanding mechanisms that we could potentially target to help delay or inhibit this transition from, say, this castration-sensitive to this castration-resistant phenotype. And so I want to also just introduce the AR gene. The canonical androgen receptor gene has 8 exons that get transcribed into an mRNA that get translated into a protein that very importantly has these 4 different domains: the N-terminal domain, the DNA binding domain, the hinge region, and the ligand binding domain. And the ligand binding domain is very critical because this is what's required for sensing the presence of and absence of androgens, while the N-terminal domain and the DNA binding domain are required for the activation of downstream target genes. And so it's been studied from Scott's lab and many others that there's been genomic deletions in patient samples as well as prostate cancer cell lines that lead to the expression of these androgen receptor isoforms, these truncated isoforms termed androgen receptor variants. And so this is just an example of one variant. This is ARV567ES. And as the name denotes, exons 5 through 7 become deleted genomically in these patients, which lead to ultimately a frame shift of exon 8 and a loss of this ligand binding domain because it's a frame shift. And you can see this could be really detrimental to these patients because now you have this variant of this androgen receptor circulating that has no ability to sense the presence or absence of androgens, but it still has all of the domains required to activate its downstream target genes. And so my talk today and my project specifically, though, is really focused on post-transcriptional regulation of the antigen receptor. So, genomically, nothing is altered in these patients, but through post-transcriptional mechanisms such as alternative splicing, or what I'll talk about today, alternative polyadenylation, there leads to be inclusion of these cryptic exon sequence denoted as CE, like CE5, CE3, and these all reside within intron 3 of AR. And through inclusion of these sequences, it ultimately leads to a very similar phenotype that I just showed you above, where you get a truncation of this transcript, ultimately lead into a loss of the ligand binding domain. This is just ARV7 and ARV9, both well-studied proteins, variants proteins. And so, as I said, today I'm going to really be showing you data, though, about how alternative polyadenylation is leading to the expression of many of these different variants. And I know many of you don't study polyadenylation, so I just wanted to very quickly introduce a basic mechanism of RNA polyadenylation. It's a critical process for the maturation of mRNAs. So there's a canonical poly-A signal, this AAU, AAA, and typically followed by the CA sequence where the cleavage actually occurs. And so, it's a very critical process for many things such as stability and export. But for the sake of my talk, and really for a lot of importance of gene expression, it defines the 3' end of the transcript. So you can see, if it's chosen more proximal or distal, it will make that encoded transcript either longer or shorter, and effectively the protein as well. And so I do want to just also introduce that this is an image from a review article a few years back. While we're very interested in polyadenylation in prostate cancer, it has been shown to have function in other disease models in cancer. Forms of muscular dystrophy, as well as other cancers, have shown that more proximal poly-A site usage is prevalent, leading to a shorter 3' UTR. And then really interestingly, a paper came out a few years back that showed widespread intronic polyadenylation in chronic lymphocytic leukemia. And this is actually very similar to what we see with the androgen receptor in prostate cancer. So, I just want to introduce this to say that while I wouldn't say it's an extremely well-studied field, alternative polyadenylation in cancer, it is becoming more and more well-known to have a role in disease progression. And so this is a pretty busy slide, but I just wanted to introduce the factors that are known to help play a role in selection of a poly-A site. I definitely do not want to go through every one of these factors. I just want to introduce these subcomplexes. There's the Cpsf complex, and specifically, I am going to show you a lot of data on Cps F1, but this complex directly interacts with this AAU, AAA sequence. There's the CSTF complex that interacts with downstream sequence elements, and then the CF1, CF2 complex that are known to bind to upstream sequence elements. And through help with Sarah Monroe at the MSI, she had really helped us determine that there is clinical evidence for high levels of specific 3' end processing factors correlating with disease progression. So this is progression-free survival looking at TCGA. And what you can see here is Cps F1, as I just showed you, as well as PCF11, show that higher expression leads to worse progression-free survival in this patient data. She also did a good analysis where she looked at enrichment in CRPC, castration-resistant prostate cancer, versus primary prostate cancer across these 4 different data sets. And what she found is, again, Cps F1 shows enrichment across all 4 of these data sets, but also Cps F4 shows some enrichment. You can see PCF11, Cps F7. So, a lot of these factors do show clinical significance, is what I want to really highlight with this slide. And I do just want to, lastly introduce the cell lines I'm going to show you, because I know a lot of you probably don't work in the prostate cancer community. I just want to make sure you know why I'm using these cell lines. And so LNCAP is a very commonly used cell line in the prostate cancer field. It's an androgen-sensitive human prostate cancer cell line that is derived from a lymph node metastasis. And so very importantly, these only express the full-length AR. No AR variants are expressed in LNCAP cells. But a very good comparison model for LNCAP is LNCAP 95, because these are actually cells that are generated from the parental LNCAP cell line, but it's just been cultured under long-term androgen deprivation conditions, which effectively make them androgen independent. They can grow without androgens in the media. And importantly, they express full-length AR, but they also now have emerged to express AR variants as well. And then the last line that I'll show data on is 22RB1. It's just another androgen-independent cell line. It's a human prostate cancer cell line that was derived from xenograft that was serially propagated in castrated mice. So it's androgen independent. And again, it expresses full-length AR and AR variants. And so these latter two lines are more of a CRPC cell line, while the LNCap is more of this castration-sensitive cell line, I would say. And so when this is work that I have done, but also work from Jamie, a previous T32 that was a postdoc in Scott's lab. When we look at this intron 3 region of AR that has all these different cryptic exons that get utilized in these variants, and we look in patient samples here, these 5 different patient samples, as well as in LNCap and LNCap 95, and I will say the top is using Pfkfb. This is a poly-A-click-seq. Essentially, it's very similar to RNA-seq, except it's very useful at determining what polyadenylation sites are being used globally. And so when we look at Pfkfb and we look within this region, with LNCap, it's an ARV negative cell line. We really see no hits for polyadenylation being utilized. But in LNCap 95, we see this one hit in this entire region right at the end of cryptic exon 3. And importantly, when we look at RNA-seq data in patient samples, we see these reads all continuing on and then all dropping off really right at this end of the CE3 site. And so to help illustrate this, I have this cartoon where, independent of this 3' splice site that's being used to generate these different variants, such as V9, or V1 even, they all seem to utilize this exact same alternative poly-A site at the end of CE3. And so Jamie, the previous postdoc in the lab, had really kind of started this project by targeting this alternative poly-A site. So she developed an antisense oligomer that would lay down right on top of this alternative poly-A sequence. And what she found is in 22RV1 cells, when you probe for AR using an internal antibody, so you detect all isoforms of AR, you completely lose expression of AR variants when you inhibit this poly-A site. And really interestingly, you actually shift expression higher to full-length AR. You can see here compared to the control. And this is quantified by qPCR on the left. You can see about a 2-fold increase in full-length AR while a decrease in all these different variants when you inhibit this alternative poly-A site. And so I do want to emphasize that it's important because we're not only reducing variants, but we seem to be actually restoring expression of full-length AR in these, highly expressive AR variant cell line. And so what I have done since Jamie's time is develop more antisense oligomers that flank either upstream or downstream of this alternative poly-A site. I showed you that slide early on of the transacting factors that are known to regulate polyadenylation. And, I was very interested if these flanking sequences also play a role in, usage of this poly-A site. And I would say that these are just denoted as roughly nucleotides away from the poly-A site. So upstream 100, upstream 50, downstream 50, and downstream 150. And there was just a very repetitive sequence here that I unfortunately couldn't target. But I do have some very interesting data. And so I did the exact same experiment that Jamie had done, except for now using more of these different morpholinos in 22RV1 cells. And what you can see here, this is the CE3 PAM that lays right on top of this poly-A site. We get that same result, an increase in full length, decrease in AR variants. But what's interesting is when I disrupt the flanking regions, upstream 50, downstream 50, we actually get a reciprocal effect. Now we're losing expression of full length AR. And we're gaining expression of these variants. And specifically with downstream 50, when you look at V7 and you look at V9, we get this increase in these variant expression, which is important because it's suggesting that this could actually be a negative regulator of usage of this poly-A site, this downstream 50 region. And I think, an important result is when I did this exact same experiment in LNCap cells, and so these are ARV negative cell lines again. And what you can see here is when I treat them with all these different antisense oligomers, when we just do a normal exposure for AR, we only get this full-length AR showing up. But when I do a longer-term exposure, we start to get this non-specific band occurring. But in the downstream 50, there's a clear doublet occurring here. And when I probe for V7 specifically, you can see that V7 is now induced in this otherwise AR variant negative cell line. And so this is really important because we're showing that there's a mix of positive and negative influence that's really regulating usage of this alternative poly-A site. And, effectively the hypothesis is that there's a transacting factor that could be binding to this region that's actually negatively regulating usage of this site. And so, of course, we're very interested in, though, what are the transacting factors, though, that are binding to this region and really driving usage of this polyase site. And ultimately, we're very interested in, do these regulate prostate cancer growth as well? And so going back to, the patient data that we had, it really highlighted Cps F1, as I kind of spoke, to be a candidate factor that we should investigate. It had high expression, leads to worse progression-free survival, and it's enriched in these 4 different data sets in CRPC compared to primary prostate cancer. And so what I did is develop shRNA. So I did stable knockdown of Cps F1 in these 3 different cell lines that I showed you: LNCap, LNCap 95, and 22RB1. And really what you can see here is across the board, when we knock down Cps F1, and I should say this is just looking at crystal violet growth, day 0 versus day 6, you can see there's a significant growth effect across all 3 of these cell lines when I knock down Cps F1. And I will say it's important because LNCap is an ARV negative cell line. So it's suggesting that it also has functions, at least with respect to prostate cancer growth, that could be AR variant independent. But I do want to say, when I looked at full-length AR and AR variants in these ARV positive cell lines, LNCap 95 and 22RB1, we do see it as a candidate regulator of variant expression. In full-length AR, when we knock down Cps F1, we see this increase in expression, and we see this decrease in B7 and B9, and this is qPCR data. And in 22RB1, we see the very similar results: increase in full-length AR, decrease in B7, decrease in B9. And this is actually very similar to the results we see when we directly inhibit that polyase site. And it's, leading us to the hypothesis that Cps F1 could be the transacting factor that's binding and regulating usage of this alternative polyase site. And I will also say that I just looked at endogenous expression of 22RB1, and this is just LNCap, again, that ARV negative cell line, and 22RB1, this more aggressive CRPC cell line. And what's interesting is when we look at Cps F1 expression, it's much higher expressed in 22RB1 than in LNCap. And this really does fit our model of, that Cps F1 could be upregulated in more advanced phases of prostate cancer. And so, with the help of Jeff Miller and the MSI, I had conducted RNA-seq analysis, doing, the exact same conditions that I showed you with the growth experiments, the shRNA knockdowns, essentially looking at either positive or negative enrichment of these different hallmark gene sets when we compare the shRNA control to the Cps F1 knockdown. And this is gene set enrichment analysis. And you can see it just sorted by its NES score, and the red ones are these significant gene sets that are either being positively or negatively regulated, or I should say, enrichment, upon Cps F1 knockdown. And so what's really interesting right away is when we look at the androgen response hallmark in LNCap cells, we actually see a positive enrichment of the androgen response hallmark upon Cps F1 knockdown. But when we look at LNCap 95 and 22RB1, we see this negative, pretty much this reciprocal response where we're seeing a negative enrichment now, and you would, it's suggesting a lesser of an androgen response when we knock down Cps F1. And, for LNCap 95 and 22RB1, this does make sense because we're losing expression of androgen receptor variants, which are more unregulated isoforms of the androgen receptor. So, we can see that there could be a lessened androgen response because of that. But it is really interesting that we see this reciprocal effect, specifically looking at LNCap versus LNCap 95. Because remember, they're isogenic cell lines, suggesting that Cps F1 could have separate function between these 2 cell lines with respect to the androgen response. And that's a really interesting, and this is definitely some very early data that we've gathered. But for the sake of the end of this talk, and what I want to continue talking about is looking at potentially AR independent mechanisms of Cps F1 regulation. And so, I'm very interested in also outside of AR, what could Cps F1 be regulating? And when we look at these 3 different hallmark gene sets, glycolysis, hypoxia, and mitotic spindle, they're the 3 consistent sets that are downregulated upon Cps F1 knockdown. And I have some really interesting data regarding glycolysis, so I'm going to really focus on that for the last part of this talk. And so, glycolysis is definitely a very complicated, long pathway. I do not want to go through all of these factors involved in glycolysis of, the glucose to pyruvate conversion. But I do just want to say that, when we look at the RNA-seq data, there is many of these glycolysis regulators are significantly downregulated upon Cpsf1 knockdown. There's GPI, which is important for this glucose 6-phosphate to fructose 6-phosphate conversion, but aldolase, GAPDH, PGK, all of them are showing significant downregulation when we knock down Cpsf1. And if I hone in specifically on GPI, this glucose 6-phosphate isomerase, we see significant downregulation in LNCAP 95. This is qPCR on the left and Western on the right. And you can see when we knock down Cpsf1, GPI is becoming significantly decreased in expression. And this factor has been really, a factor of interest to me, because when we look at our PAC-seq data, so I, on top of RNA-seq, performed a PAC-seq with the exact same setup with the shRNA knockdown, the 2 different shRNAs and the control shRNA. And again, just to remind you, PAC-seq is very similar to RNA sequencing, except for the hits really pile up exactly where polyase site usage is occurring. And so what we can see here, it's kind of hard to see. This is a UCSC genome browser. This is the canonical end of the GPI 3' UTR. And so it's kind of defined as the canonical polyase site. And in my control samples, you can see there's usage of these 2 sites right here in this canonical 3' UTR. But, really interestingly, when I knock down Cpsf1 here, we're seeing now induction of this more distal polyase site. It's much beyond the annotated 3' UTR. And so this is suggesting that a 3' UTR lengthening event is occurring in GPI upon Cpsf1 knockdown. And so I just want to end that, this section with the, very speculative model. I want to emphasize speculative because we have a lot of stuff still to test, but we're really excited by this on how, Cpsf1 could be regulating GPI. And so in the Cps1 high conditions, or the unaltered conditions, as I just showed you in the control sample, we really just see the canonical polyase usage in this GPI 3' UTR. But when we manipulate Cpsf1 expression and it becomes low, we see that a subset of these transcripts now appear to be using this more distal polyase site. And our hypothesis is that this is leading to a 3' UTR lengthening event, which could be causing the destabilization of GPI, say through microRNA or just other destabilizing elements that only exist in this more lengthened 3' UTR, which ultimately leads to this decrease in GPI expression, which could lead to an ineffect to process glucose 6-phosphate to fructose 6-phosphate. This is that step that GPI is important for in glycolysis, and ultimately lead to this growth effect we're seeing with Cpsf1 knockdown. And so with that, I just want to conclude what I have talked about today. In the first part of the talk, we really nominated this polyase signal, this alternative polyase signal in CE3 of AR to be a driver of AR variant expression. But also we found that when you inhibit this site, you can actually restore full-length AR expression in prostate cancer cell lines and decrease AR variant expression. And also when I inhibit the flanking regions of the site, you actually lose expression of full-length AR and increase expression of ARVs. We've shown you that there's clinical evidence for Cpsf1 in progression-free survival. There's higher expression of Cpsf1 leads to worse progression-free survival. And Cpsf1 is enriched in these 4 different data sets in CRPC versus primary prostate cancer. And lastly, as I just showed you some data on, when I knock down Cpsf1, we increase full-length AR in prostate cancer cell lines, we decrease AR variants, we decrease cancer cell growth across all 3 of these cell lines I've tested, and glycolytic enzymes also, as I just showed you, show lower expression. And so, we're really excited by a lot of this recent results. And I just want to end the talk on some of the things I'm currently doing right now in the lab. And, most importantly, I want to determine what is the mechanism of GPI regulation by Cpsf1. And also, what is the therapeutic significance of disrupting GPI in prostate cancer? And so just going back to this model, key questions we have to address to really solidify this model is, first and foremost, do GPI transcripts decay more rapidly upon Cpsf1 knockdown? Also, is there a buildup of 6P when we knock down Cpsf1 because there's a lack of GPI to help this conversion? And then very importantly, if we treat cells with known GPI inhibitors such as ERA4P or PG, does that phenocopy now these growth effects we see by Cps F1 knockdown? And then lastly, this is something we're still very interested too, and this is what I introduced kind of early on in the talk is what is the therapeutic significance of this alternative poly-A signal in AR? I've shown you that this site is critical for AR variant expression. I didn't have time to get into it today, but AR variants are proposed to be resistant to drugs such as enzalutamide because they lack these domains that are targeted by enzalutamide. And so a hypothesis would be, if we can target this site in CRPC models, could we now resensitize them to, say, anti-androgen treatments? Because now we're restoring full-length AR. And really importantly, we're interested in doing this in vivo as well. If we make mouse PDX models, can we inhibit now, say, growth of these PDX models when we inhibit this site? And so with that, I just want to acknowledge the entire Dehm Lab. Scott's been a really great mentor for this project. Everyone else has helped a lot with support and advice. And Jeff, Sarah, and Rebecca and the MSI have all helped contribute and Rendong on this project as well. And I'll take any questions you guys have.
Thanks, Kyle. That was really great. Amazing. Stephanie Huang has a question. Is the poly-A site variant mutually exclusive to other AR variants like the exon 567 deletion? If not, what is there interplay among these different AR variants?
So I would say from a lot of our data that I showed early on with the sequencing, it does seem that independent of the variant that's used, this poly-A site really is utilized. So like V7, V9, V1, all of them utilize the same poly-A site. Now I've shown that when we inhibit this site, pretty much every variant has an effect. And I should say not every variant, because when I introduced the ARV567, that's an actual genomic deletion that's that's causing that expression of the variant. And V1, V6, V7, V9 are all these splicing outcomes that are arising in these variants, is what I would say. So kind of a short answer would say all of the ones that arise from, say, splicing or alternative polyadenylation do use the same poly-A site.
Great. Sergio Gradoloni has a question. You can see this. Fantastic talk. I have a very naive question. If we forget about prostate cancer, these alternative AR poly-A sites are normally there. So perhaps one would ask what the normal physiological role of these variants might be. Do they express sometime during development or are then silenced? Are they expressed in any stressful conditions? I think these are excellent questions.
Yeah, that is a really good question. And to my knowledge, these, I think that there's some belief in the field that these could be arising from a more aggressive form of prostate cancer. So of course it's that, question also, is this arising because of the cancer becoming more aggressive, or is it a driver of the prostate cancer progression, I would say. And to my knowledge, I don't think we detect these variants in primary prostate cancer. I think it is, because the whole hypothesis is that these variants are helping confer resistance in these really more aggressive prostate cancer cell lines.
So, I mean, these factors like Cps F1, I mean, you had several, right? Are they ever co-expressed or developmentally expressed? Like maybe a way to look at that might be to see if the factors that regulate the usage are expressed developmentally or in stressful conditions or in early stage prostate development. I mean, something like that. If there's public data, that might be easier to track. You know what I mean?
Yeah, and I definitely, I've looked at. So I will say, these 3' processing factors. The thing is that they're critical for all RNA maturation. So they are expressed pretty much everywhere, I would say. And I haven't looked at developmental stages of, say,
look in brain.
Yeah, but it does seem like the data I've shown that in more aggressive like prostate cancer, like CRPC and 22RB1, that we actually see higher expression. And actually, interestingly, in NEPC prostate cancer, we actually see very high expression as well. But yeah, I haven't looked at developmental, like early to later phase expression. But my hypothesis would be that these would all be expressed kind of consistently because they are general 3' processing factors. It's more of a hypothesis that maybe these become hijacked and get higher expression in the prostate, in more aggressive cancer.
Cool.
Yep.
Great. Well, I just want to make a quick announcement. Any postdoc can present in this venue. It does not have to just be T32 associated postdocs. We're looking for anyone who's ready to go. So let me know and we could schedule that. And then I just want to comment about postdocing for a long time or any stage of your career. I postdoced 7 years also. And one, it was really fun and I don't regret a moment of it. And two, and probably more importantly for my career, it really gave me a lot of savvy, much more savvy because of the career development activities and the networking and learning all the political stuff that goes along with these academic careers. And it really has served me well. So I didn't mean to in any way downplay. You should never feel bad about a long-term part of your training. I think you should embrace that because you're going to get so many rewards in so many ways that you won't even know what those are until they happen. So again, I want to congratulate these two fabulous scientists about their work and thank you for wonderful talks today. And hang in there. We'll get through COVID together and be well. Thanks a lot, everyone.
Thanks.